This podcast will deal with meiosis. And when we talk about meiosis, I like to ask, what is the advantage of sex and what is the purpose of meiosis? Now, if you remember mitosis, the purpose of mitosis is to produce genetically identical daughter cells. Remember that equation, 1 to the 46th power equals 1. On the other hand, the purpose of meiosis is to produce genetically diverse daughter cells, the daughter cells here being the gametes. Remember this equation, 2 to the 23rd, 8,388,608 possible combinations from independent assortment of homologous chromosomes at metaphase 1. And this number, it gets even way larger because of homologous recombination or crossing over that occurs in prophase 1. This diagram compares and contrasts mitosis and meiosis, and I'm going to spend a little time going over it because it might help to put things in perspective. So let's look at somatic cells, mitosis on the left, and meiosis, what's occurring as gametes are formed. So in mitosis, one cell division results in two daughter cells. Chromosome number, humans 2n equals 46, and so you have a single cell division producing two daughter cells, 2n equals 46. Here, if we look at DNA replication, there's a pre-mitotic S. DNA goes from 2C to 4C and S, and then through G2 and M, and back down to 2C during mitosis, and back to the 2C DNA content. There's normally no pairing of homologous chromosomes, so you don't form tetrads. There's normally no recombination. This is an important point. During metaphase of mitosis, there's bi-orientation of sister kinetochores. There's loss of cohesion between sister chromatids during metaphase, and so the centromeres divide at anaphase, and each pole gets an identical copy of the DNA from each chromosome. If you look at meiosis, on the other hand, in the cells that give rise to gametes, there are two cell divisions that are going to result in four products of meiosis. So you're going to start with a cell that is 2n. It's going to go in the first meiotic division to n. The second meiotic division also is going to go to n. But here you're going to look at differences in DNA content. We can look at a pre-meiotic S phase for both cell divisions. DNA replicates, so you're going to go from 2C to 4C. First meiotic division, you're going to go 2C, and the chromosome number is also reduced. The second meiotic division, there's no DNA replication. The chromosomes divide a second time. DNA content is reduced, so you're going to go from 2C to 1C. The chromosome division in the second meiotic division is much like a mitotic division. So here we look at what's happening in meiosis 1. There's full synapsis of homologous chromosomes in prophase. A tetrad forms, so these are the homologous chromosomes. Let's say this is, for example, maternal chromosome 1, paternal chromosome 1. Remember, there are 23 pairs in humans. Here, showing some crossing over. Here, the chiasmata, the point where crossing over has occurred. In the first meiotic division, there's co-orientation of sister kinetochores towards the same pole. There's maintenance of cohesion between sister chromatid arms during metaphase 1 of meiosis. So at the first meiotic division, an entire homologue goes to one pole. So here both chromatids of maternal chromosome 1 go to the one pole. Both of paternal chromosome 1 go to the opposite pole. This produces variation in gametes. So here's another way to look at it, comparing mitosis and meiosis in humans. In mitosis, there's one DNA replication, one chromosome division. So if you look at for humans, 2C is 7 picograms, 2N is 46 chromosomes. So you go from 7 picograms to 14 picograms through S. So you're entering at the end of G2 14 picograms, 46 chromosomes. Go to G1, you go through division, back to 2C, 46 chromosomes. DNA amount remains the same, chromosome number remains the same, and this is because the chromosomes are behaving as individual units. Meiosis, there's one DNA replication, there's 2 chromosome divisions, so that a diploid cell produces 4 genetically different haploid cells. Notice what happens here. Pre-meiotic DNA synthesis, you go from 7 picograms to 14 picograms, 46 chromosomes. First meiotic division, you go to 7 picograms of DNA, 23 chromosomes. Second meiotic division, 3.5 picograms of DNA, still 23 chromosomes. And it is because at metaphase 1, the chromosomes are behaving as homologous pairs. They're synapsed with each other. So just to remind you, mitosis 1 to the 46 is 1, meiosis 2 to the 23rd, a really big number. Meiosis in males leads to an even distribution of cytoplasm among sperm. Meiosis in human males begins at puberty, and it can actually continue through the 7th or 8th decade of life, and meiosis in human males reflects a renewing cell population. We'll talk about this in a little more detail as we talk about the male and female reproductive systems, but here you've got a spermatogonium, which is a diploid cell. It's 2N2C. The primary spermatocyte is 2N4C. At meiosis 1, the secondary spermatocyte is 1N2C. At meiosis 2, the spermatid is 1N1C, so the secondary spermatocyte is haploid with respect to chromosome number. The spermatid is haploid with respect to chromosome number and with respect to DNA content. And in sperm maturation, there's a series of post-mitotic maturation events that occur to get mature sperm. We'll look at that in much more detail when we talk about the male reproductive system. The female system is unusual. Meiosis here in females leads to an uneven distribution of cytoplasm between the ovum and the polar bodies. As you probably know, meiosis in human females begins at 3 months of fetal life, and it then arrests, and it remains arrested until puberty, and then at puberty meiosis resumes, and it continues through menopause. We talk about meiosis in human females as reflecting a decaying cell population, and we'll talk about this in more detail as we talk about the female reproductive system. But here you've got, for example, oogonia dividing by mitosis. They're 2N2C. Eventually they're going to enter the primary oocyte stage at about 6 to 7 months of fetal life. They'll be 2N4C. The LH surge just prior to ovulation stimulates the first meiotic division, and that will produce a secondary oocyte, which is 1N and 2C, and a polar body. Now, these are drawn out of proportion because, of course, the polar body is much smaller than the secondary oocyte. Meiosis 2 in humans will only occur if fertilization occurs, and at meiosis 2, the secondary oocyte will divide into an ovum, which will be 1N and 1C, and a second polar body, which will also be 1N and 1C. The first polar body may divide. It usually in humans disintegrates. Meiosis 2 is occurring as fertilization occurs. The sperm actually has to penetrate the egg for the second meiotic division in humans to be complete. Once that second meiotic division is complete, then the pronuclei will fuse and you'll begin to get the zygotic divisions. We'll talk about this in much more detail when we talk about the female reproductive system, but meiosis is clearly under endocrine control, and this is just to put that in perspective for you. I'm not going to go over it in detail here because we will go over it in detail in the female reproductive system, and you clearly go over it in much more detail in your physiology course as well. Just some points to wrap up about meiosis versus mitosis. In mitosis, chromosomes align as separate entities on a metaphase plate. That's what's shown here. In meiosis, homologous chromosomes pair and align as synapse, bivalents, or tetrads on the metaphase plate of meiosis 1. So, for example, let's compare these two figures. This would be paternal chromosome 1, maternal chromosome 1. They align separately in mitosis. Maternal chromosome 2, paternal chromosome 2. They align separately in mitosis, so during metaphase, each sister chromatid goes to opposite poles. Here in meiosis, the homologous chromosome synapse, so maternal chromosome 1, paternal chromosome 1 align as a homologous pair. Maternal chromosome 2, paternal chromosome 2 align as a homologous pair. So here in humans, there are 23 homologous pairs on the metaphase 1 plate, whereas in mitosis in humans, there are 46 individual chromosomes aligned on the metaphase plate. Metaphase 1 of meiosis, the homologous pairs separate, so an entire chromosome consisting of both sister chromatids goes to one pole or the other. This figure shows the tetrad or bivalent, consists of 4 chromatids. So these would be the maternal chromatids, the paternal chromatids, the chiasmata, the point of crossing over between the homologous chromosomes. Chiasmata are essentially a manifestation of recombination that actually occurs in zygotine or pachytene. You don't see the chiasmata until much later in prophase 1, sort of at diplotin or diakinesis. This just shows the 46 human chromosomes, 22 pairs of autosomes, and the pair of sex chromosomes, so 2n equals 46, and they're just displayed nicely on the right side of the figure. This figure finally compares and contrasts mitosis and meiosis again. The first meiotic division, we call it the reduction division. DNA content goes from 4C to 2C. Chromosome number goes from 46 to 23 at the first meiotic division. The second meiotic division, we call it an equational division. It's similar to mitosis. DNA content goes from 2C to 1C. Chromosome number remains at 23. And I will leave it to you to look at this figure and try to consolidate in your mind the differences between mitosis and meiosis. With respect to the stages of meiosis, we talk about a pre-meiotic S phase where DNA synthesis occurs, and then meiosis 1, the reduction division, sometimes called the heterotypic division. Prophase 1 is very protracted, it's very elaborate, and in human females it can last for decades. But there's a leptotene stage where there's a condensation of chromatin and assembly of lateral elements of the synaptonemal complex. The zygotene stage is the assembly of the central elements of the synaptonemal complex. The synaptonemal complex, of course, is the structural basis to allow homologous recombination to occur. That recombination occurs in pachytene. Then you have diplotin or diakinesis, and in females there's a decondensation of chromatin here, and you have an intense amount of transcriptional activity, especially for ribosomal RNA. You're going to have translation of proteins that are specific proteins needed for the egg to survive. Then at metaphase 1, there's going to be recondensation of chromatin. Transcription and translation is going to cease. In human females, this is only going to occur if ovulation occurs. Then there'll be anaphase 1, telophase 1. There's a very brief interphase after telophase. There's no DNA synthesis. In humans, you can't hardly recognize that interphase. Meiosis 2 is the equational division or the homotypic division. In human females, this only occurs if the egg is fertilized. So there'll be a very quick prophase 2, metaphase 2, anaphase 2, then telophase 2, and in sperm, after telophase 2, there's a whole series of maturation events that are going to occur as the sperm is going to mature. In the egg, after telophase 2, the pronuclei will fuse, and then you'll get into the first embryonic divisions. This final slide is to remind you that meiosis uses two mechanisms to mix up or shuffle genes, and that's going to lead to genetic or biologic variability. The diagram on the left shows independent assortment in meiosis to produce 2 to the n different combinations of haploid gametes. So this would show a cell that potentially has 6 chromosomes, 3 homologous chromosomes. You can, I think in your mind's eye, do the pairing and come up with 1, 2, 3, 4, 5, 6, 7, 8 possible gametes. 2 to the third, 3 homologous pairs, 8. It'd be a little bit hard to do that in humans, but 2 to the 23rd combinations, as we said, well over 8 million. And then the diagram on the right showing homologous recombination in meiosis 1 is exchanging segments on homologous chromosomes. It's sorting the genes. This diagram shows 2 crossover events on 1 pair of homologous chromosomes. The 2 crossover events there would lead to 4 possible gametes. So here you can imagine, combine these numbers by multiplication. 8,388,608 possible combination of gametes times ever how many crossover events occur. You're going to multiply that number times that number. It's an infinite number of possible gametes that can occur as a result of meiosis, thus assuring that no gamete is the same, thus assuring genetic variability, thus an absolute necessity for the evolution of species. Finally, the last slide, this internet film from McGraw-Hill. It's got a nice, simple animation to show how meiosis works, but you'll see as you click on that video, the animation will come up.